Hello there, and welcome to this Khan Academy tutorial on AS level biology. In this video, we will look at cell immunity. This is the final part in the immunity series where we'll look at ELISA tests and the pathogenesis of HIV. So let's get started. Here are the aims of this tutorial. We will look at what an ELISA test is, describe the types of ELISA tests with named examples, and look at how viruses infect cells and cause disease, and explain how HIV causes disease. And finally, we'll look at why viruses are difficult to treat, and especially HIV. So, ELISA is short for Enzyme-Linked Immunosorbent Assay. These tests allow you to detect presence of specific antibody or specific antigen. There are two types of ELISA tests. One is direct, which uses one antibody, as seen in this image. Another one is indirect, which uses two different antibodies. Note that antibody that binds directly to the antigen is called the primary antibody, while the secondary antibody binds to the primary antibody. So let's first look at indirect ELISA. In this example, we will look at identifying if an individual has antibodies to the HIV virus. Firstly, the HIV antigen is bound to a well plate. Then, the patient's plasma is added, because plasma contains antibodies, but it does not contain blood cells, which may interfere with the results. If the individual has HIV-specific antibodies, this will bind to the HIV antigen, and we call this the primary antibody as this directly binds to the antigen, as I showed previously. The well plate is then washed to remove unbound antibodies, as again this could interfere with the results. This is followed by adding secondary antibodies specific to the primary antibody. You can see this from the diagram that this antibody binds to the constant region of the primary antibody. Furthermore, you can see that an enzyme is attached to the secondary antibody, whose function we'll come on to. Again, the well is washed to remove unbound secondary antibodies, which may provide invalid results. Next, a solution containing the enzyme substrate is added to the well. If the secondary antibody is still present, the well changes color, as the enzyme converts the substrate to a colored substance. This color change would indicate the presence of HIV antibodies in the patient's blood. Now let's look at the direct ELISA, and here we'll use the example of the pregnancy test. The pregnancy test detects the presence of the hormone human chorionic gonadotrophin, or HCG for short. This hormone is measured in the urine of a woman. The application area is where urine is added, and it contains mobile HCG antibodies with a colored bead. If the urine contains HCG, it will bind to the antibody, forming an antigen-antibody complex. This complex moves up the test strip and binds to the immobilized antibody complementary to the HCG hormone. Remember that all antibodies used so far here are primary because they bind directly to the antigen. The immobilized antibody contains an enzyme that changes the color of the bead attached to the other antibody, indicating the presence of HCG. If no HCG is present, the mobile antibodies move along the strip and bind to complementary immobile antibodies in the control window to show that the test has worked. If there are two lines, the woman is pregnant. While two lines are often an enjoyable moment for any lady, the same can't be said for COVID lateral flow tests, which work in a very similar fashion to the pregnancy test. Now, let's shift gear to look at viral pathogenesis. Pathogenesis means how a pathogen causes disease. Firstly, scientists still debate whether viruses are truly living or non-living. But, in general, viruses infect cells by binding to receptor protein on the host's cell surface. Once in, the virus breaks the host cell's DNA and uses the host's amino acids to synthesize new viral proteins. And this is because viruses have no organelles. When many copies of the viruses are made, eventually cell lysis occurs, i.e. the cell breaks down and eventually dies. A virus to learn further about in A-level is HIV, which stands for Human Immunodeficiency Virus. This is a type of a retrovirus, which means it converts RNA to DNA. HIV does this by using the enzyme reverse transcriptase. The genetic material of HIV is RNA, which is inside a capsid protein. Let's understand how HIV infects cells. The attachment protein of HIV binds to the receptor of a T helper cell, and the capsid is released into the cell, and the HIV RNA is converted to DNA by the HIV's enzyme reverse transcriptase. The DNA made is single-stranded because an RNA is a single-stranded molecule. So, in order to make a double-stranded DNA molecule, we need to use another enzyme called DNA integrase, and this is already present in HIV. Once a double-stranded DNA is made, it is inserted into the T helper cell's DNA. Hence, HIV's DNA is copied many times, making many HIV molecules. Eventually, the T helper cell is killed by cell lysis. Finally, let's look at why viruses, and specifically HIV, are difficult to treat. Firstly, viruses are intracellular, meaning they are more sheltered from the immune system. Imagine hiding inside a room than being in open ground. Very difficult to find, right? Moreover, it's difficult to produce drugs that target only infected host cells, as they are not often easy to identify. Hence why vaccines are a better alternative. Remember, the vaccine is not a treatment but reduces the risk of severe symptoms of the virus. Also, antibiotics don't work on viruses, they only work on bacteria. And antibiotics tend to work by targeting the cell wall synthesis in bacteria. Now, HIV is more difficult to treat because it infects the immune cells themselves. Killing your own immune cells is a bit like killing a member of your own family. Not that you would, but oh god, this tutorial's becoming more and more insidious. But anyways, even if our immune cells try to kill T helper cells, the HIV does a better job of it anyways. HIV eventually makes the person so immunosuppressed that they develop a syndrome called AIDS, which stands for Acquired Immunodeficiency Syndrome. Ultimately, individuals with AIDS don't just die of HIV, but often due to another common diseases such as a cold, which is caused by the virus influenza, and other common diseases. And this essentially worsens their chance of survival, and hence AIDS is truly a devastating condition. Phew, that was a long tutorial. Not my longest, but if you stayed this far, well done. Here's a recap of the points we covered in this tutorial. As usual, use them as questions to consolidate your learning. I thank you for watching, and I'll see you in the next one.